Good morning, students. Today, let me continue with that chapter digestion, and today's topic is mechanism of digestion. Digestion is a process of the conversion of complex food materials into simpler molecules which can be readily absorbed and assimilated by the cells of the body. And this physiology of digestion involves two main aspects. One is mechanical digestion, and the other one is chemical digestion. The first one is mechanical digestion. As the name itself suggests, it is also called physical digestion because only the physical treatments will be given to the food materials that we consume. And this mechanical digestion involves the following aspects. The first one is mastication or chewing. This mastication or chewing, it occurs in the buccal cavity, and it is aided with tongue, teeth, and saliva. And once the food is converted into tiny bits, that food will be swallowed from the mouth cavity towards the pharynx and then towards the esophagus. And this process of movement of the food materials from the mouth cavity or the buccal cavity into the pharynx, we call it as swallowing or deglutition. Actually, that converted food mass that is produced in the buccal cavity, it is called as bolus or boli. And the second step of mechanical digestion, it occurs in the esophagus region, what we call it as peristalsis. So peristalsis is nothing but a simple wavy downward movement of the food mass from the esophagus towards the stomach. And you should have aware of one thing, that in the esophagus region, there are no digestive glands, so there is no chemical digestion. So only mechanical digestion takes place in the esophagus. Then the third step of mechanical digestion, it occurs in the stomach region, what we call it as churning. So churning is nothing but a process of conversion of food materials that the stomach has received, and that will be mixed up with the gastric juice, which contains so many aspects of the contents in it, and it will be given a kind of movement which we can compare with that of a mixer. So this process is called churning, and the end product of that mechanical digestion that occurs in the stomach, we call it as chyme. And this chyme is acidic in nature, and that chyme will move towards the small intestine region, that is towards the duodenum. And the fourth step of the mechanical digestion, it occurs in the small intestine, the whole part of the small intestine, what we call it as the segmentation movements. And the segmentation movements, as the name itself suggests, it is the part by part flow or the movement of food mass in different parts of the small intestine towards the large intestine. So these are the four basic steps involved in mechanical digestion, and let us go through such details here. The first one is digestion in the buccal cavity. In the buccal cavity, both mechanical and chemical digestion takes place, and the teeth chew the food into smaller bits, and these bits are pushed and rolled and tasted by the tongue. The food mixes with the saliva in the buccal cavity. The water in the saliva dissolves the food. The mucus in the saliva lubricates the food, and the lysozyme in the saliva kills the bacteria, and the enzyme taline or salivary amylase converts the starch into maltose. Thus, in the buccal cavity, only the carbohydrate digestion takes place because there are no proteases in it and there are no active lipases in it. The food is then swallowed by the action of tongue and pharynx, and the swallowing action rotates the food into small globules called the bolus or boli, which I already mentioned. The boli travels through the esophagus through a process called peristalsis, the definition of which I already told you. It is a downward wavy contractions of the wall of the esophagus, and thus the food reaches the stomach, and its process of swallowing is also called deglutition. Coming to the digestion in the stomach also both mechanical and chemical digestion takes place, and the walls of the stomach contract and relax, mixing up the food with gastric juice. The water of the gastric juice dissolves the food, the mucus lubricates the food, and the HCl present in the gastric juice kills the bacteria, loosens the fibers, releases the iron, activates pepsinogen and prorenin. The pepsin converts proteins into proteoses and peptones, and the enzyme renin converts the milk into casein and whey. And the casein combines with calcium ions to form calcium caseinate. The product is also known as curd, and the process is called curdling. Then the calcium caseinate is converted by pepsin into proteoses and peptones. The enzyme lipase in the gastric juice may convert fat into fatty acids and glycerol, but this action is doubtful since the fat has not yet been emulsified in the stomach, but rather it occurs in the intestine with the participation of bile juice. The intrinsic factor of the gastric juice helps to absorb the vitamin B12, and the partially digested acidic food in the stomach is called the chyme. And this chyme slowly passes down through the pyloric sphincter into the duodenum, and in the stomach mainly protein digestion alone takes place. The reasons are, in the stomach, that is in the gastric juice, there are no carbohydrates, and even though the gastric lipase is present, but the amount is very less and the fats are not yet emulsified, hence no lipid digestion, but only protein digestion occurs in the stomach. This you have to remember. And this protein digestion, as I already told you in the earlier part of this slide, it is aided by the two active enzymes called pepsin and renin. In the duodenum, when the chyme comes to the duodenum, it mixes with the secretion of the Brenner's glands, that is of the intestine, and the liver and pancreas. The Brenner's gland secretion changes the acidic chyme into alkaline chyle, and the secretion of the liver is the bile. It contains no enzymes, but it contains bile salts, and bile salts like sodium taurocholate and sodium glycocholate, and it contains bile pigments like bilirubin and biliverdin, and the bile salts emulsify the fats, provides alkalinity to the chyle and kill the bacteria. The bile pigments helps in detoxification by excreting the toxins. So this is the role of bile juice. Coming to pancreatic juice, the pancreatic juice contains sodium bicarbonate and several enzymes in it. The sodium bicarbonate helps to neutralize the HCl, and the enzyme trypsin converts proteins into polypeptides. The enzyme carboxypeptidase converts the polypeptides into di and tripeptides. The enzyme amylopsin converts polysaccharides into disaccharides. The enzyme steapsin converts the fat into fatty acids and glycerol. So you should remember one thing here at this juncture, that steapsin is the only active lipid digesting enzyme present in the human stomach or in the human alimentary canal. The pancreatic juice helps to digest all kinds of food, namely carbohydrates, proteins and fats. So it is only digestive juice which can digest all the types of food that we consume. And from the duodenum, this alkaline chyle passes into the jejunum, the next part of the duodenum. In the jejunum, the food mixes with succus entericus, that is intestinal juice, and the maltase of the succus entericus converts the disaccharide maltose into monosaccharides, namely glucose. The sucrase converts sucrose into glucose and fructose. The lactase converts lactose into glucose and galactose. So, these are the three main enzymes present in the intestinal juice, which plays an important role in the digestion of disaccharides mainly. Hence, these three enzymes like sucrase, lactase, and maltase, they are referred to as disaccharase. The enzyme erepsin converts di and tripeptides into amino acids. Nucleosidases converts the nucleotides into purines, pyrimidines, and pentose sugar. And the lipase converts fat into fatty acids and glycerol. The phospholipase converts phospholipids into fatty acids, glycerol, and phosphoric acids. The non-digesting enzyme of the succus entericus, namely the enterokinase, activates pancreatic trypsinogen into trypsin. So you should remember one thing here, that this enterokinase is the only non-digestive enzyme present in the human digestive system, and its role is to activate pancreatic trypsinogen into trypsin. It acts as an activator only. The hormone secretin stimulates the pancreas, and the intrinsic factor helps to absorb the vitamin B12. Thus, in the jejunum, almost all processes of digestion gets completed. Carbohydrates are hydrolyzed to form glucose, proteins are hydrolyzed to form amino acids, and fats are hydrolyzed to form fatty acids and glycerol. The digested food passes on to the ileum, where the absorption takes place. So these are the activities of digestive system in the jejunum region. In the ileum, so in the ileum, actually no digestion takes place because it has already got completed in the previous part, that is jejunum. And in the ileum, the main process concerned with digestion is absorption. The inner walls of the ileum are provided with 1 mm tall, minute finger-like projections called the villi. And these villi increases the area of absorption up to 600 times. And each villus is covered by a layer of tall columnar epithelial cells with microvilli. And below the epithelium, there is a network of blood capillaries, and at the core of the villus, there is a minute lymphatic capillary called the lacteal. And the glucose and amino acids from the lumen of the ileum passes into the blood capillaries due to diffusion or osmosis, while the fatty acids and glycerol passes into the lacteals with the help of bile salts. Thus, all the nutrients enter the circulatory system and get distributed to the tissues, and this happens mainly in the ileum region. Coming to the structure of a villus, the details of which I already explained you in the previous slide. So this is the structure of a single microvillus. As you see in the picture, in the central part there is a lacteal, which is a part of the lymphatic system or the lymphatic vessel, and surrounding which you will be able to see the artery, you will be able to see the veins, and surrounding to that you will see this finger-like projection called villus, and this villus has many smaller hair-like processes called microvilli, and in the upper part of this common basement membrane, you will happen to see the presence of columnar epithelial cells, which are the main cells involved in absorption. And this is what one structure villus. And this region, which is present at the base of two successive microvilli, actually almost it comes here, there it will be having a glandular projection called crypts of Lieberkühn, which contains intestinal glands in it, and these intestinal glands they secrete the digestive juice called intestinal juice or succus entericus, which plays an important role in digestion, the details of which I have already explained you in the previous slide. Coming to the digestion in the colon, the undigested and unabsorbed waste from the ileum comes to the colon or the large intestine, and in the colon the excess water, salts and glucose are reabsorbed. Thus, the fecal matter becomes solidified. The cellulose digesting symbiotic bacteria present in the colon help to digest the cellulose in the fecal matter to a certain extent. Then, the fecal matter is lubricated by the mucus secreted by the inner wall of the colon, and the feces then reaches the sigmoid colon, where it stays till the time of defecation. The yellow color of the feces is due to the presence of a pigment called stercobilin. So, let us go through the summary of these digestive processes, which I already explained you in the previous slides, but let me summarize as follows. So, in the buccal cavity, salivary glands are present. They secrete saliva, and as I told you already, it contains only amylase, that is salivary amylase, the only enzyme, and it is also called ptyalin. And this salivary amylase, it converts the complex polysaccharides into disaccharides like maltose through various forms of dextrins. Then fats are being digested by lingual lipase into fatty acids and glycerol, but the amount of this lingual lipase is very negligible, though they are present, and the process of emulsification has not yet taken place, so there is no fat digestion strictly speaking in the buccal cavity. Then in the stomach, as you know that gastric gland secretes gastric juice, and the complex proteins are they are acted upon by pepsin and HCl to produce the products called proteoses and peptones. And this milk contain a protein called casein, and that casein is acted upon by renin, and that will produce a product called calcium caseinate or calcium paracaseinate. The product is called curd, and the process is called curdling of milk, which I have already told you in the previous slide. And this calcium paracaseinate, it is acted upon again by pepsin, and that gets converted into proteoses and peptones. And coming to fat digestion in the stomach, though the lipase is present, but it is not a sufficient amount, and also the emulsification has not yet taken place. So this is the summary of digestion process in stomach region. Now coming to intestine, that is the first part, duodenum. Duodenum receives all the three digestive juices, that is from the liver, bile juice, and from pancreas, pancreatic juice, and from intestine, intestinal juice. So that is why some people or some authors, they call this duodenum as Sangama of digestive juices, as it receives the digestive juices from the liver, the pancreas, and small intestine. Coming to fat digestion, in the duodenum region, it receives the bile juice, which contain bile salts, and those bile salts converts the complex fats into emulsification. So emulsification is nothing but the process of conversion of complex fats with high surface tension to produce simpler fats with low surface tension, and the products of this emulsification of fats is also called the micelles, and these micelles are tiny cells or the tiny complex globules of fat molecules. And these bile salts, when they acted upon food, they provide alkalinity to the food mass. And toxins, if present in the food, they are detoxified and they are excreted through a mechanism what is called as detoxification. Then, in the duodenum, pancreatic juice is also enters, and the sodium bicarbonate present in it neutralizes the HCl. And the protein present here in this duodenum region, it is acted upon by trypsin, and that converts it into proteoses and peptones. And the enzyme chymotrypsin, it acts on proteoses and peptones and converts it into polypeptides, di and tripeptides. And the polypeptides, they are acted upon by carboxypeptidases, which gets converted into di and tripeptides as well as into amino acids. And the polysaccharides are acted upon by amylopsin, and that is converted into disaccharides. And the only fat digestive enzyme called steatopsin, and that acts upon the complex fats and converts it into fatty acids and glycerol. So you should remember one thing, that before the steatopsin acts on fats, they have to be emulsified, and that has been brought about by these bile salts. Then, in the summary of digestion process in jejunum region, that is with the help of intestinal juice, that is succus entericus. As I told you already, enterokinase, it converts or activates trypsinogen into trypsin, and the di and tripeptides are acted upon by erepsin to produce amino acids, and the nucleosidases acts on nucleosides and producing purines, pyrimidines, and pentose sugar, and the disaccharases like maltase, sucrase, lactase, they act upon their respective substrates like maltose, sucrose, lactose to produce the glucose, fructose, and galactose respectively. And the fats are acted upon by lipase to produce into fatty acids and glycerol, and the phospholipids are acted upon by phospholipase to produce fatty acids, glycerol, and phosphoric acid. And the hormone secretin, it activates the pancreas to produce its digestive juice. And the intrinsic factors, they are activating for the absorption of vitamin B12. So this is the summary of digestion process in the jejunum region.